Okay, female reproductive system. Now, two things before we start. Okay, put on my serious face. All right? Everybody put on your serious face. We're in college. We're going to be using words and terms that in high school everybody giggled at when we talked about it. We're not going to start giggling about boy parts and girl parts. Okay? The second thing is, I said this on the very first day of class, and I really have to strongly reiterate it. In our society today, there's a lot of different genders. So when we're talking about female reproductive, this is biology, okay? Pure, simple biology. Male reproductive is biology. Okay, are we all in agreement that we're talking about biology today? Okay, all right. So this is female reproductive system. And what the reproductive systems are for is really continuation of the species. We wouldn't be here without reproductive means. So what does this do? Sexual maturation, especially when we're talking about the gametes. You're going to hear that word a lot. The gametes are either the egg or the sperm in humans. Those are your sexual cells, or some people consider them the germ cells. Reproductive system, you got to have a means with which you get the sperm and the egg together for fertilization. And for the female reproductive system, it's for the maintenance, protection, support of the developing embryo and the fetus. Now we got to go back a little bit and talk about some cell stuff. I promise it's not going to be painful. How many people are familiar with mitosis? Did we do mitosis in a MP1? Yes. Okay. What is the four-letter thing we say when we talk about mitosis? PMAT. Those are the stages. And so this is going to happen in our body cells. This is how we grow. This is how we repair damage. And you start with one cell and you end up with two identical daughter cells. Basically, this is cloning. Cells just cloning themselves. Now, this is nuclear division, but we also divide the cytoplasm as well, cytokinesis. So just a quick review. Did you do meiosis in AMP1? Okay, that's a little bit trickier, and some of you may not have had it, so that's what we're going to talk about today, because meiosis is how we get the gametes, how we make the sperm and the egg. Because when you think about it, how many chromosomes do we have? Why does that say 23 and you say 46? You have 23 chromosomes from your dad, and you get 23 chromosomes from your mom. You have two pairs of all your chromosomes. You got two chromosome number 1s, two chromosome number 2s, and your 23rd pair is your sex chromosomes, either XX, biological female, XY, biological male. That's the 23rd pair. So in mitosis, you start with one cell that's got 46, you end up with two cells that have the identical set of 46. Here, we've got to divide the genetic material. Because can you take a 46 chromosome egg and a 46 chromosome sperm and have a baby? No. That's not compatible with life. We want the baby cells to have 46 chromosomes. That means the male gamete has to have 23 and the female gamete has to have 23. So in this meiotic division, we have to have the number of chromosomes. What does that mean right there for our starting cell? 2N? Twice the number. Pairs of N is the number of chromosomes. We call that diploid. Ploidy are the number of chromosome sets you have in your cells and organisms. Your body, your somatic cells, your body cells are diploid. Now, what did we say the four letters were for mitosis? PMAT. Meiosis, you're going to do PMAT twice. And in the first PMAT, we are going to produce two daughter cells that lose the 2 and they only have one set of chromosomes each. Then we're going to go on to divide, and we're going to have 4 daughter cells that have half the set of chromosomes. So where we reduce the number is in meiosis 1, that's the reduction division, and then meiosis 2, we're simply dividing again. Now we're going to get into a little more detail about how we do this. Do we know the difference between a chromosome and sister chromatids? You remember that difference? Your chromosomes are linear. They just look like your finger. What do sister chromatids look like? It's your chromosome duplicated but held together at the centromere. Those are the sister chromatids. Now, when we look at going through these divisions, all of our chromosomes in our cell are going to become sister chromatids. That means we have 46 sister chromatids. 2 of them are going to be sister chromatids that represent both of your chromosome 1s. Another pair are going to be both of your chromosome 2s, one from dad, one from mom. And what we're doing in this first division is we're separating and migrating chromatids that are still together with their pair into a daughter cell. So here we have 23 sister chromatids together, those X's. And in this division, now we're separating the sister chromatids, and we've just got chromosomes in each of the 4 resulting daughter cells. Okay? You following me so far? We got some more pictures because this is important. I see people going, no, you lost me at chroma what? Okay. And then there's our PMAT1 for the reduction and PMAT2 for the just simple separating of the chromatids. All right, so here's a picture. I like pictures better anyway, right? Mm-hmm. So here we have our normal somatic looking cells, right? 1 chromosome, blue from dad, pink from mom. We'll say that's our 2 pair of chromosome 1s. Since we have our chromosomes paired, they are called homologous chromosomes because they have the same genes represented on that chromosome 1. The alleles for that gene might be different. Do you understand when I talk about genes versus alleles? You have the gene, let's just say for instance, I don't know it's on 1, but just for an example, on chromosome 1 you have the gene for eye color. You got to have the gene for eye color on there. But dad's eye color gene may be the allele for blue. Mom's eye color gene may be the allele for brown. You see, alleles are what flavor is the gene. Okay? But these are homologous chromosomes. So we replicate. When do we replicate into chromatids in our cell cycle? Do you remember? S phase. So we got interphase, G1. What does the G stand for? Growth 1. Synthesis, the S phase, is when we replicate. Then we've got growth 2 and on and on and on. We're going to have the same thing happening with our PMAT for meiosis. But here's our mitosis. We have our homologous chromosomes. All 46 line up at the equator. When do we line up at the equator? PMAT, metaphase, M. And then we separate them at anaphase and end up with 2 resulting cells that are identical to what we started with. That's mitosis. Meiosis, we're going to take a little branch off here, and the first thing we separate are our homologous chromosomes. Those first 2 daughter cells are going to have 23 of our sister chromatids together, representing every one of our essential chromosomes. The other is going to have the different 23. That's going to be happening as we separate these, and then we end up with, what do we got here? Meiosis 2, where they each have 1 chromosome. I wish they had kind of left these together, but this is anaphase of meiosis 1, where they're pulling them apart, pulling the sister chromatids apart here, and you're going to end up with 4 daughter cells. Does that help a little bit? Got to get homologous chromosomes, sister chromatids, chromosomes. Again, it's kind of back to that confusion that you have in AMP1 when we first start doing this with mitosis. It's going to be important we understand that difference as we move forward. So again, homologous chromosome are the pairs. They've been replicated, so since they're replicated, although they're homologous chromosomes in name, they're really sister chromatids because they're attached to their copy. And then here we can see we're pulling them apart.
What makes it homologous again?
What makes it homologous? The same numbered chromosome. You know, we have 23 pairs, and we're going to see a picture that shows how different they are. So homologous chromosomes are your pair.
2 and 2.
Yeah, you're 2 and 2. One from mom, one from dad of 2. One from mom, one from dad of 3.
Okay.
You know, if you were to take chromosome 1 from dad and chromosome 4 from mom, they're not homologous. They're not the same.
Okay.
Okay?
That makes sense.
The same as in genes, not the alleles. The alleles can be different. Yeah. This is the picture I was talking about. If you were to look at a cell and you look at all the chromosomes and they've labeled these for some genes, you see the banding pattern here. You can take that banding pattern and transpose that into like a computer program, and do you see how we've matched the banding pattern on these and the length? And the same for those. You see how they got 1 and 1, homologous, 2 and 2, homologous, 3 and 3. These are your homologous pairs all the way down to your X and your Y chromosome. Notice Y doesn't do a lot. Kind of like guys in general, right? Okay, I'm sorry. Work harder, not smarter, right? We got the homologous? Say what?
Nothing.
Well, I didn't quite hear that, but here's the thing. We're going to talk about development at the end of the male reproductive chapter. We got the egg, we got the sperm. We're going to talk about development. Early in development, your default pathway for biological gender is female. That's the default, female. Your reproductive systems are going to go through an indifferent period where male, female is and looks the same, including the genitalia. The Y chromosome is here to do two things. One, first and foremost, turn off the female chemical signals and to turn on the male signal. If you don't turn off the female, then you're going to get one of these hermaphroditic conditions where you have a blend. I mean, it's kind of rare, but not incredibly rare if that happens. But it just goes to show female is the default gender. Now, we don't do this in humans, but I believe reptiles and alligators, whether it's a female or a male, depends on the temperature of the eggs in the nest. And they can develop either into a lot of males or a lot of females.
Bio 2 study we had to do of that.
Yeah, I mean, it's crazy some of the things that happens in the animal kingdom. Doesn't happen for males. It's all about the X and the Y chromosome controlling these chemical signals and growth factors. So again, we've already talked 23 equals 46. We did mention that meiosis is going to reduce the number of chromosomes. Because you can't have 46, you got to get it down to 23 because you're going to get paired with another gamete with 23 to get the total number for the next offspring. The second thing that happens in meiosis, and not second in order, but just another thing that happens, is called recombination. And when I say recombination, you do not necessarily have the same chromosome that either your dad or your mom had. Now, you're getting half from mom and you're getting half from dad. But remember, they got half from your grandmother and half from your grandfather, right? And so what happens when we take our homologous chromosomes, one from one parent, one from the other, as we're going through this process and these chromosomes start to condense in prophase 1, we've already replicated them. We did that in the S phase of interphase. Now at prophase, we're condensing the sister chromatids down, and sometimes their arms get tangled up. It's not an accident. There's a mechanism for it, and it creates a cross, which is called a chiasmata. And that chiasmata doesn't just overlap, but it causes the exchange of portions of chromosomes. And this process, because there's a crossing over, that's the name of this type of recombination, crossing over. So you can imagine at the end of meiosis, there's going to be one gamete that's got just this chromosome, this all light colored, one that's got this complete darker color, but then we're going to have one gamete that's got this mix-up, and we got another gamete that has this other mix-up. Do you see the recombination that can happen? So your chromosomes may be a blend of your mom's and your dad's, a combination that never existed before, which is part of what makes you a very unique individual. Now we got something else that makes you pretty unique. Well, here, look, here's our homologous chromosomes. Here you can see how they've crossed over, the sister chromatids. There's the exchange happening in anaphase. And here we're beginning to pull them apart, right? Because now we're in anaphase 2. In that process of crossing over and the separating of our homologous chromosomes is referred to as independent assortment. In metaphase of mitosis, how many of our sister chromatids were lined up at the equator to be pulled apart? In mitosis, 46, all of them. Because we got to have 46 chromosomes going this way and 46 going this way. In meiosis, you've got the homologous chromosomes lined up, stacked up in pairs. So one homologous chromosome, again, these sister chromatids still, one's going to go to this daughter and one's going to go to this one. So let's say chromosome 1. Our cell up here gets mom's. The cell down here gets dad's. Okay? What about chromosome number 2? Who's going to go which way here? Is mom's going to go up or mom's going to go down? Completely random. 50-50 shot. It's like having a boy or a girl. It's a coin toss, 50-50 shot. If you've had 4 girls and you get pregnant again, what's the chance you're going to have another girl? 50%. Unless you're me, I had 3 girls, I would have a 100% chance of having another girl. Just destined, no. Just destined, all right? So it's kind of like statistics. And when you look at this independent assortment, you can basically say, okay, since we got a 50-50 chance of going in either direction, and we have 23 different homologous chromosomes, the number of combinations that you can generate, 3.4 million different combinations.
So that means you're gonna have like 3.4 million kids, and they all be.
You better not have 3.4 million kids. Whew.
They'll be like,
That's how many gametes you have. But here's the thing. How many egg are going to produce a baby? How many sperm are going to help produce a baby? Okay? So there's going to be one gamete, a female and a male, that's going to come together.
How many did you say?
8.4 million. What did I say?
we said 3.4.
I said 3. I'm sorry, I was seeing the 3. 8.4 million, a lot. Okay, just a lot. That's my favorite scientific word, a lot. And stuff, that's another one. But here's the thing. Even with that many combinations of our homologous chromosomes, all of our homologs are not going to be the same because of crossing over. So now, if we're talking about instead of 2, make that a 4. We're getting to the point there's almost infinite number of different combinations. And that's why you may look kind of like your dad, maybe a little bit like your mom, maybe kind of like your sister or your... That's because you're similar, but you're very unique in your own way. And this variety, this is what is so important in survival of the species. When you look at farmers that are growing commercial crops, they don't all have the same strain of a crop year after year after year after year. Because if you do, and they're all resistant to the same bad stuff, but here comes the bad stuff that wipes everything out, all the crops are gone. I think we thought that was what was going to happen with COVID. Since we're all very similar, this one horrible disease come along, it would wipe us out. But the variability of the species, genetically speaking, helps prevent that. It might get a lot of us, but it's not going to get all of us. Make sense? Understand? It's so very important. Now, we're going to get to male next. I just put this picture to show that for meiosis, you're going to start with one primary cell, a stem cell to give rise to the sperm. And in the end, you're going to have 4 spermatozoas. These are the actively motile ones once they get to the female reproductive tract. And they're each going to have a different set of those chromosomes, 23 pair. That's meiosis, right? You start with 1, you end up with 4. Trick question. For female gametogenesis, do you start with 1 and end up with 4?
Yes.
No. You get shortchanged, ladies. Well, not really. So, in the fertilization process, what do you get from dad? What do you get from dad at fertilization? The sperm, but what goes into the fertilization to cause this new organism, the zygote, the embryo? What comes from dad? 23 chromosomes, and that's it. You don't get anything else from dad. Dad's just going to give you the bare minimum. Does that sound familiar? You ask dad for money, is he ever going to give you more than you asked for?
Nope.
What? You spoiled? Spoiled? You notice all the ladies are going, yeah, more every time. Guys, do y'all get more? We don't get any. No. Go get a job. So you get 23 chromosomes from dad. What do you get from mom? Everything else. You get the 23 chromosomes, but you get the bonus of all the Golgi, all the mitochondria, all the endoplasmic reticulum, all the ribosomes, all the cytoplasmic stuff has to come from mom. And a little preview of development after fertilization, that one fertilized egg begins to divide into a number of different cells, but these first generations of divisions, there's no growth after the division. So you got to start with a pretty good sized egg. And to do that, as we go through meiosis, you start with that parent cell, okay, that's going to produce the 2, I'm doing air quotes, daughter cells. No, all the cytoplasm is going to stay with 1, and only a membrane-wrapped set of 23 chromosomes is going to go to the other. It's called a polar body, and it's waste. And we're going to do the same thing. So with females, you're going to end up with 1 egg, and you're going to throw away the first polar body and the second polar body. See, this one's not even going to make another branch to give you the 4. You're going to have this one large egg with all the stuff that fertilized egg is going to need to make a baby, and all you're going to get from dad is just 23 pair of chromosomes. But it's kind of important to get those 23.
Aren't women born with like all the eggs that they'll ever have?
Yes, you are born with all the eggs you're ever going to have, and you have over 1 million to start with. By the time I think you get to puberty, it's down to, a measly 400,000.
What? Don't we lose an egg every single time we have a baby?
More than. Yeah, we're going to get there.
What?
Yep, we're going to get there. You're going to learn all sorts of stuff about your reproductive system you didn't even know. Hopefully. No, you do. It's important to know. Okay, so the female reproductive organ where we find the gametes is the ovary. They're paired. You have a right and a left. And with our ovary, we're going to have a fairly tough capsule around the ovary. We've even got a connective tissue band that's kind of white in color. It's called the tunica albuginea. and alba is white. Like albinism, white, no pigmentation. You have this line that's right down the middle of your body called the linea alba. Do you remember that one? Yeah.
The germinal epithelium and the tunica albugina are the same thing?
No. See the white, the light color there? The germinal epithelium and the capsule is going to be on the outer part. And then again, we got cortex and medulla, right? Cortex, outer, medulla in the middle. The cortex is typically where we're going to see the earliest of our oocytes, the eggs, and their cells that are around them that make them into a follicle. We're going to find those scattered throughout the cortex, and you can see some of these follicles in various stages of maturation. So we're going to go through that process as well. So here, follicle equals oocyte plus the cells of the follicle, some call them granulosal cells, and together that package makes up your ovarian follicle.
Just that little circle right next to the ovarian.
There's a lot of them that they're not illustrated in here. But we're going to talk about all the different kinds of follicles. So, you first have all of these very, very, very early follicles that are going to have your oocyte, the egg, surrounded by one squamous cell layer of follicular cells. And those are called primordial follicles. That's where you have 1.5 million, and then you've got 400,000 still viable by the time you hit puberty. And this is going to be when you haven't even been born yet, that 1.5 million. Now, as these follicles mature, the cells of the surrounding cells around the granule, they are going to get taller and be more like cuboidal cells. And these are going to begin to secrete the hormone estrogen. Estrogen and progesterone, we're going to see, are critical in your ovarian cycle. There are a couple of different places where you're going to get the contribution of estrogen and progesterone. Now, your primary follicle, I want to point out some things, and you'll see these in some of the histological slides we have. As this develops, you're first going to have a single layer of cuboid cells, or your follicle. That makes it a unilaminar primary follicle. Uni means one, right? Unilaminar, one layer. In this picture, you can see there are multiple layers of these cells. That's going to make it a multilaminar primary follicle. As this gets bigger and bigger and you produce more of these follicular cells.
Is that the only thing that secretes estrogen? Like that's
No, there's more. This is just one of them. All right?
What is meant by like primary oocyte?
So that's the egg. So as you know, here before you're born, that oocyte has started going through meiosis, but it's held in suspended animation in prophase 1. It's condensed, it's got the sister chromatids, but it's just on hold. And some suspect that these oocytes, eggs, held in that prophase 1 state maybe isn't as stable as your somatic cells being held in a certain state. With increasing maternal age, you see congenital birth defects. And what's the one you probably hear about the most? Down syndrome. The older you get before you have children, the greater the risk of having a baby with Down syndrome. Now there's also evidence that the older the male is, that happens as well, but not as drastically as it does with female maternal age. Yeah?
So do these like type of follicular cells or follicles all coexist, or is this like a spectrum, like it starts off as primordial?
Well, yeah, we're going to see that all of these existed for a long time. You're going to see some of these as they begin the maturation process, Every ovarian cycle you have, there are going to be a number of follicles that begin the developmental process, but there's only typically one that ovulates per cycle, and it usually bounces back and forth from one ovary to the other. You usually don't ovulate from the same ovary at once, or 2 months in a row. The follicles that began to develop but didn't ovulate just simply regress. They're called atretic follicles, and they just get resorbed. But you can't use them anymore. That's why you got 400,000, but you're not going to have 400,000 babies, or 3 million babies. 8.4 million.
So when you're ovulating, what is happening to the cells?
We'll get there.
Oh, okay.
You're getting ahead of me.
I'm sorry. You
That's
keep
all right.
talking about it, and I'm like, okay, what's happening?
Yeah, we'll get there.
I feel like this is an old question.
No.
But when females are on birth control, does that just pause the cycle?
Oh, that's a
Now, for many women, it does. It depends on the hormones and your type of birth control. I believe that there may be other birth control pills, because these are hormone replacement medications typically. And what we're going to see is that these hormones also control the lining of your uterus. And so by the end of this, you may be shocked that anybody ever gets pregnant. The timing of having a baby is redonkulous. The egg exists for 24 hours after ovulation. The sperm only survive 48 to 72 hours. They have to fertilize in exactly the right place in the fallopian tube, or it's not going to work. So some of these hormones may also impact the lining of the uterus. So if fertilization were to happen, it can't implant. But I know a lot of women, once they get on birth control, they don't have a period. Especially if they're older, premenopausal type women. They just don't.
Can't women have children in their uterus?
Yeah, and we'll get to that. Yeah, we'll get to that. Great questions. Great questions. We'll get there. Okay, let's work with our follicles here. Okay, some terms that we need to know. So for our secondary follicle, these are getting older and older in the maturation process. All right, they were stimulated by hormones 1 month, and they're starting to grow. And what happens is some of our follicular cells, they start to produce a fluid. And it's one of the most enjoyable words that I say in biology, because your tongue just kind of likes to bounce around. This fluid is called liquor folliculi. Say it, liquor folliculi. It sounds like some on Sesame Street or something. Liquor folliculi. You can't say it without smiling. Liquor folliculi. Liquor. Yeah, like liquor. Yeah. Liquor folliculi. And this fluid starts to get produced by these follicular cells, and they start to pool in these spaces and start to make the follicle grow bigger. And all of this fluid is eventually going to collect into one large fluid-filled central chamber that is called the antrum. And what eventually is going to happen, and we're going to see this, well, let's not get there yet. We're going to see this in the next slide that we're coming up. But basically what this is going to do is it's going to make a big lake of liquor folliculi. And the egg, here in the middle, the oocyte, that's going to be the nucleus that you see there in the circle. And that white band around the oocyte, that is called the zona pellucida. It's acellular, it's filled with proteins and carbohydrates. It's one of the fertilization barriers for the sperm. But you also see some of these follicular cells still attached around the zona pellucida and the oocyte. You see that? Those cells are going to stay there, and those cells are called the corona radiata. You know what corona means? Not beer. Crown. And the radiata is it radiates around the oocyte and the zona pellucida. So the corona radiata is another fertilization barrier that the sperm have to get through. So this lake of liquor folliculi, once we get to this stage, this is going to be so big, and this unit of the oocyte, zona pellucida, and the corona radiata is going to detach, And it's going to be free floating in the antrum filled with liquor folliculi. That is referred to as, your book says, vesicular follicle. I've always known it as a Graafian follicle. I'd probably put Graafian. If it was fill in the blank, I'd accept either, but for me, I've just always known Graafian.
Do what Mr. Dudley does and give us bonus points if we get both names.
Well, I wouldn't put both. If I put both names, then it would be incorrect. Now, what's going to happen with ovulation is this one follicle is going to win the race. There are going to be a number of these Graafian follicles in your ovary developing at the same time, but whoever wins the race gets to ovulate. And that egg, zona pellucida, and corona radiata is the one that's going to get ejected into the fallopian tube. So the liquor folliculi, those granulosal cells pumping this stuff out, that follicle is going to get bigger. It's 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 going to get as big as your whole ovary. When you ovulate, is it kind of not comfortable? Can you kind of feel a pinch when you ovulate? Some people say they can feel the pinch at the point of ovulation. Because how does the oocyte, how does it get out of the ovary? It bursts. It's like pumping up a tire and you put too much air in it and it just pops. The hydrostatic pressure of that liquor folliculi inside this one Graafian follicle that's as big as your whole ovary, it ruptures through the capsule of the ovary and it gets shot out into your lower abdominal space. The fluid, the oocyte, zona pellucida, and corona radiata, it gets ejected. And if you feel a pinch, that's why, because you're ripping through the ovary.
What are cystic ovaries?
Cystic ovary, these are some of these follicles that develop inappropriately and you don't get the ovulation, but they get really big and it's uncomfortable and it completely jacks up your hormones in your body. It's a big problem, I think, for a lot of people. It's called polycystic ovary syndrome. It's not the leading cause of infertility, but it's one of the causes of infertility. Because with PCOS, polycystic ovary syndrome, you have no idea when you're going to ovulate or not. There's not a regular cycle. 2, with PCOS, when you do have cis rupture, now instead of just liquor folliculi, you've got a lot of these cystic cells, these follicular cells that also get ejected as well. And a lot of that debris gets into the fallopian tube and it clogs it up. It's not endometriosis, it's just a blockage of the fallopian tube. So even if you were to ovulate, the sperm and the egg can't get together. So that's some of the issues.
Whenever it bursts through, does the fluid go out with it?
Yes.
And does it like harm us or like?
No, it just gets reabsorbed.
Okay.
No, you made it. Yeah.
Okay, so during ovulation, it's
Now we're going to cover ovulation again down the road, but
it's known to have the time, so like the best time for you to have a baby.
Mm-hmm.
So does that, during this stage, does it let off like a bunch of hormones?
so we're going to talk about all the hormones that happen that causes ovulation. It's hormonally controlled,
Oh, okay.
as is the development of the follicles.
I have an earlier question. It's about the primary oocyte.
Uh-huh.
With the zona pellucida, is that like a capsid or a capsid on a virus?
You can kind of think of it like that, but it is just a spherical shield around the oocyte that the sperm have to use enzymes to penetrate through. Some of the zona pellucida have protein receptors that the sperm attach to that causes them then to release the enzymes to basically eat their way through the zona pellucida to get to the membrane. And typically it's going to take more than one sperm to get through all of those layers, but once one sperm gets through the membrane, that's it, game's over.
But there's still proteins.
Yeah, proteins, carbohydrates, yeah.
Okay, so if I got this right, the vesicular, that follicle right there is the largest one that
Yep.
like women have. And so
Yep.
that's the one that does start. Like it's the first one that is to be ovulated.
The first one that ovulates ends the race. You may have two that are pretty close to the same size, and the first one that shoots out the oocyte, the other one just goes,
Yeah, they have the biggest oocytes. So,
the oocytes are same size.
oh,
The
it's
oocytes,
whatever
it's the
surrounds them.
follicle,
The
yes,
follicle around them.
yes.
So it's only one egg?
Yep,
'Cause-
one egg gets out. Sometimes you can get more, it's how you get twins.
So like you said, if there's like two of them that are like neck and neck basically for when they start the race.
Somebody's got to win the race. If they both do it at the same time, it's possible, then you could potentially have twins.
What facilitates which one is released?
Just random, whoever has the higher pressure from the liquor folliculi and whoever ruptures first. Yeah. Yes, ma'am. They just regress and are reabsorbed. They're called atretic follicles. You can see the cells dying, and they just get reabsorbed into the ovary tissue.
That's sort of my question. I was like, if two are like basically neck and neck and one goes first, is the woman just going to be in pain for the next month?
No, it's just like, they just regress. Yeah. It's pretty magical. Okay. So after ovulation, the follicular cells from that ovulated unit, they're not just, okay, game over, I'm going to go home. No, those cells become a corpus luteum. And those granule cells, they start making progesterone and estrogen. So here's where we're beginning to get more of that in this cycle. And that's going to continue for maybe up to 10 days to 2 weeks after ovulation. But if you don't get a zygote, now in your notes and on the PowerPoint slides, I think it says if the egg is not fertilized, if you don't get a zygote, that implants. That's how that should read. If you don't get implantation of a zygote, then after 2 weeks, this corpus luteum is going to run out of gas. Your progesterone and your estrogen start to decline, and that's when you have your menses. That's when you shed the uterine layer and you start over. So that's what's controlling, keeping the uterine lining ready for a possible implantation. Now, after the corpus luteum runs out of gas, then you basically have a scar tissue-like unit, and this is called a corpus albicans. What is alba? White. It's just kind of whitish looking scar tissue that happens inside your ovary. And that, of course, also gets removed and reabsorbed as you go along. Because this is happening every month. Hopefully not the same side every month. You give this ovary an extra month to heal while you ovulate over here the next month and back and forth.
What would happen if it didn't do that?
I guess it can. I guess you could ovulate from both at the same time. It just keeps on going.
Is it possible for an unfertilized egg to implant on the uterine lining?
No. And we'll see why. And it might be towards our developmental. That's when we're going to get to the fertilization part. What causes implantation? It's not just the egg sitting down on the uterus. There's a whole lot more to it than that. All right, let's look at some pictures. Look at some pictures. These are histological views of an ovary in cross-section. So in this case, here is our capsule, our germinal epithelium. Can't really see our linea alba, maybe a hint of it here. But you see how all this looks kind of mottled and speckled looking out here? All of those are your primordial follicles. There's a lot of them. And you can see as we go deeper in the cortex, getting close to the medulla, you can see how they develop in the various stages of development. We have one here that's almost completely detached. And this is getting close to a Graafian follicle. And this whole thing with those corona radiata cells, the zona pellucida and the oocyte, will be free floating out here in the antrum that's filled with the liquor folliculi.
The follicle is the whole thing.
The whole thing, the oocyte, the granular cells, the whole thing, that's the follicle.
Okay.
Is there like an ideal follicle, like a sperm cell would go to?
Oh, sperm cell's not going to the follicle because this is what the sperm cell's going to when it's in the fallopian tube.
So, it goes in the fallopian tube
Yeah, and we'll see a picture of one in just a couple of slides here.
Wait, I thought it ruptured.
It does, it hasn't ruptured yet.
No, it hasn't. That's why I said in the fallopian tube, just that unit.
Yeah, so whenever the egg travels down, it's popped and then it.
The egg doesn't pop, the follicle pops. Yeah, 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 yeah.
Around the egg when it pops through.
The zona pellucida and the corona radiata cells, that's all that's around the egg. And those are kind of protecting it, but they're also the fertilization barrier. Yeah. So here we're looking at the capsule, our germinal epithelium. These, you see how we've got our squamous cells here? So those are primordial follicles. This is a nucleus, that's our nucleolus. And you see how the chromosomes have begun to condense? That's prophase 1 of meiosis, and that's where all of these are held in that suspended animation. Okay? So those are primordial. Lots and lots. Here's some more primordials, but now as we go deeper, do you see how this follicle is bigger? And those cells aren't flat, but they're more cuboid? That is our primary follicle. That's a unilaminar primary follicle. Here we have a unilaminar primary follicle, but these cells are a little bit taller. What's going on with this one over here? There's no oocyte. What you have to keep in mind when you're looking at histology is the plane of section. This thing is big enough and round enough, the oocyte may be out this way and you cut it off when you were preparing your specimen, or it's so deep you can't see it. That doesn't mean there's not one there. For your follicles, there's always going to be an oocyte present. You just may not cut through in the right plane to be able to see it.
At the very bottom of the last slide, is that like a giant one?
Don't know.
No.
Probably, but we're going to look at bigger and bigger ones. Like this one. This one is a multilaminar primary follicle. What would make it a secondary follicle? Having some of that liquor folliculae, those pools, and we don't see any of those pools out here. So we've got a lot of our follicular cells. That pink band, that's the zona pellucida, and that's the oocyte inside, and there's the nucleus of the oocyte. Now, notice these cells out here are doing something a little bit different. These are going to be a lot of those leftover cells that don't go with the oocyte that are going to be pumping out the estrogen and the progesterone. These are called thecal lutein cells, and that's not something that you got to put down, but these are going to be the ones pumping out the estrogen and progesterone after ovulation.
So is luteinizing hormone?
We're going to get there, yeah. We're just looking at all the follicles and everything, then we'll get to the hormonal control. So here we have another follicle, single cell, so that's a primary unilaminar. We got multiple layers here, but we have a big space for our liquor folliculae, so that's secondary at least. We don't know if it's Graafian or not because we can't see where the oocyte is in this particular slide. But look here, we're starting to get those spaces. Do you see those white spaces? Those are filling with the liquor folliculae, so this is an early secondary follicle. This is a close-up. We've just about completely detached from the follicular wall. And so this is what is going to be ejected at ovulation. You've got your corona radiata cells, maybe 3 or 4 cell layers thick. The zona pellucida is in here, and then there's our oocyte cytoplasm and the nucleus. That is what's going to be ejected and starts floating down the fallopian tube to be fertilized. And the sperm have to get through the cellular layer, the acellular zona pellucida, and penetrate through the membrane to fertilize the egg. Okay? That's an entire ovary. And you can see 1, 2 follicles. Now, this one, do you see how this is starting to get folds in here? That is a corpus luteum. So that antrum with all of that fluid in it, once it's released, the follicle collapses and all of those thecal lutein cells that we saw, they're all going to be in here pumping out the estrogen and the progesterone. This is probably going to be one that's just regressed and is in the process of regressing because you don't see those folds like you see here with the corpus luteum. You see all these others that were in this fairly late stage of development, those just have to be reabsorbed. So you see multiple ones begin the process every cycle. That's why you start with 400, well, you start with 1.5 million, but you have so many because you're expending several every cycle.
So where's the egg in this one?
Oh, the egg's gone. We've already ovulated, and that's the corpus luteum. And this is a corpus albicans. That's the scar tissue one. And here we have 2 other fairly large follicles that are in the process of regressing. The ovary's pretty tough. I mean, the ovary has to be tough to do any of this kind of stuff. All right, now let's get to the hormones. The hormonal regulation of follicular development and ovulation. Is anybody surprised at how much the endocrine system does in its regulation of the body? We said the endocrine system controls about half of your functions in the nervous system, another half. Well, we're coming back to our good friend the hypothalamus here. And the hypothalamus begins this process by secreting gonadotropin-releasing hormone. So what do you think this hormone is going to cause to be released? Gonadotropins. And what are our gonadotropins? FSH and LH. Follicle-stimulating hormone and luteinizing hormone that come from the anterior pituitary.
Interesting.
Which we learned about. Is that the first chapter we learned about this semester? We've come full circle now, haven't we? So follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone sort of tells you what it's doing, right? Stimulating the development of the follicles. Luteinizing hormone is also going to contribute to the point of ovulation. So when we look at this, we're getting all these productions. Our gonads are also going to be producing hormones, right? Our follicular cells producing estrogen and progesterone, corpus luteum cells producing these as well. So we have all of these controls happening. The gonadotropin release, LH, FSH, estrogen, and progesterone. These are all playing critical roles in this sort of dual cycle, not just follicular maturation and ovulation, but also the cycle of the uterus, because they're intimately tied together. Now, I know we said we're going to do fertilization maybe Thursday or next Tuesday, but I'm going to go ahead and introduce this point. Fertilization has to occur almost in the end of the fallopian tube. Your ovary sitting here, and when ovulation happens, that package of the oocyte and the granular cells and zona pellucida are pulled into the opening of the ovary. There are these little finger-like projections that kind of surround it to help guide it in. You have cilia creating a current and contractions of the uterine tube that sort of, with peristaltic contractions, create this current to go toward the uterus. But you have to have fertilization almost at the end. Not at the very end, but there's a major portion called the ampulla, and fertilization has to occur there. Because that fertilized egg going through those cellular divisions, it has to be a zygote in the neighborhood of 6 or 7 days old, because it's producing these cells, and I say that term loosely, but it's this collection of cells on the outside that have the receptors and the mechanisms to attach and to burrow down into the uterus. That's implantation. Now, I like to say that the zygote, the implanting zygote's like a tick. It's worse than a tick. A tick just stays on the outside. These things burrow down into the lining of the uterus, and all that's left behind is a little kind of pimple on the surface of where it went in. So if you don't have a fertilized egg, the egg will never be able to attach or implant. It just goes in there and it comes out in the menses.
Because it doesn't develop those things.
Correct. It's called the syncytiotrophoblasts, but you didn't hear that. Do you have a question?
You answered.
Okay. Syncytial trophy. He was going to ask about the syncytial trophy. Okay. So here's the cycle. So first, FSH and LH. You can see our cycle. We're going to start day 1. You can see that under the control of gonadotropin-releasing hormone, we produce follicle-stimulating hormone. That's going to dominate. But then as we go further along in our cycle, you can see luteinizing hormone, follicle-stimulating hormone dips below luteinizing hormone. Now, that's going to coincide also with our follicular development. And so as we're getting into this vesicular or Graafian follicle stage, remember, our follicular cells are going to be making more and more and more estrogen. Well, estrogen, and once we get that peak of estrogen, guess what? That's going to cause this surge of luteinizing hormone. You get a little bump here of the follicle-stimulating hormone. The surge of estrogen leads to that spike that's going to cause ovulation. Luteinizing hormone, follicle-stimulating hormone, they largely get turned off. And you can see as estrogen declines, progesterone is going to start taking over. And it's going to be this combination here you see toward the end of the estrogen and progesterone at fairly high levels for this last 10 days to 14 days. That's what's going to keep the endometrium maturing and being at its fullest to be able to prepare for implantation. Now, in this particular illustration, we didn't fertilize the egg. We don't have a zygote or an implantation happening. So the estrogen and progesterone, they play out, and poof, we get menses. You see how the layer becomes really thin again. We shed this functionalis top layer, and we just keep the regenerative basal layer, and we get ready to start it all over again.
So the functional layer is what gets shed?
Yes, the functional layer is the portion of the uterus that sheds every month. That's the menses. Yeah.
Does the level of hormones determine the length of the actual menstrual cycle or the shedding? Like some people it's 3 days, some people it's
Yeah, I don't think it's the amount as much as it is the timing. Because if you were to, spread this out further, then maybe the timing is going to be off more. You've got to get to a point where you have the rapid increase. I don't know if a slow increase would do the same thing. So I don't necessarily know how that would impact any particular cycle per se. I don't know, and again, I have no personal experience, but having 3 daughters, a couple of them have the PCOS. That's how I'm familiar with what's going on with that. But it seems like, by and large, most cycles are fairly consistent. And y'all are weird because when y'all get together, y'all synchronize your cycles, and it freaks us guys out.
Do you know how that happens?
I don't.
The moon.
I'm afraid to know.
It was a full moon last night.
I always wondered, having 3 daughters and having a wife, so 4 women in the house, I was like, okay, do I want them all 4 to synchronize their cycle and just get it all over with in 1 week, or just have them do 1 a week and just be that way all my life? I would pick the once a week for my whole life, because I think if they all did it in that same week, they would kill me in my sleep.
Yeah.
Because y'all are crazy, or we think y'all are, during that 1 week.
So for those people that don't have a regular cycle, what happens to that layer, that functional layer, since it's not being shedded?
If it's not shed, it's going to continue to build up. And if you're not regularly shedding that functionalis layer and it continues to build up more and more, potentially some of that functionalis layer that's just going to keep dividing and dividing and dividing could make its way into the fallopian tube. And what is that condition when that happens? Endometriosis. Because once it gets into the tube, you don't have that junction between functionalis and basalis, so you can't shed it out of the tube. It just stays in the tube, and it just continues to grow and clog up the tube even more and more. And I think that's one of the leading causes of infertility, endometriosis.
Okay, makes sense.
So,
Yeah, actually, women in the hospital with PCOS who like go to the hospital for those pains.
Yeah.
Say it's like awful.
Oh yeah, it's apparently no fun. Because you've got nerve endings in the fallopian tube and it's not supposed to stretch. You've got muscles that can, but once it starts filling up too much, it's no good, no bueno. Here's another change in the notes, make sure you get that. If the zygote doesn't implant, I think it says if the egg is not fertilized. If the zygote doesn't implant, that's when you're going to shed the cycle because the corpus luteum plays out. We're going to see with fertilization, if you get implantation by that 10 to 14 days as the corpus luteum is supposed to play out, you've developed the structures to implant, and some of those cells and some of those structures signal the corpus luteum to keep going until the placenta is large enough to make its own.
Make its own what?
To make its own estrogen, progesterone. Okay. So, this leads me to right what I was saying. If you have fertilization and you have implantation, this zygote, this implanted zygote, you can see this happening right here. Those little purple fingers that's sticking out, grabbing on, that's the syncytial trophoblast. It's the beginning of the placenta. Well, the placenta is going to start making human chorionic gonadotropin, HCG. Ever heard of that? No. HCG.
What starts secreting it?
This is going to be the zygote, one of the cellular components of the zygote. Just put it that right now. But it's going to be the beginning of the placenta. And that HCG then feeds back to the corpus luteum to keep it making estrogen progesterone. That is going to keep the lining of your uterus from shedding until the placenta is able to make its own progesterone and estrogen. That is a corpus luteum. That's the rest of the ovary. Notice this right here. It's filled with blood. When your ovulation happens and you rupture through the capsule, you're going to rupture blood vessels. And so blood will fill into that empty antrum. It was just ovulated. And before you get a corpus luteum, you can get, this is hard to say, a corpus hemorrhagicum. Hemorrhage?
Yeah.
Hemorrhagicum, and it just fills with blood, and again, that regresses too.
So when you get like hyperstimulation whenever you do like IVF and they go in and pump it,
Yeah.
you get all those things. Is that what fills? Because I thought it was the liquid that was inside goes into like your abdomen, but is it the
Now, if they're going in and they're removing the oocyte, then you probably don't have a lot of the blood.
Okay, so it's just the liquid that's inside the liquor.
Liquor folliculi, yeah.
What is
Yeah.
this?
What is what? This is the ovary, and this is a corpus luteum. I don't know why it looks like it's lobed like that, but that's a corpus luteum. That's making this estrogen and progesterone for that 10 to 14 days. This is supposed to be inside the ovary, and maybe it's just kind of lobed how it's done. I don't know. But that's part of the ovary. How big? I don't know. How big is your ovary? It's not as big as your fist. Yeah, I mean it's yeah. I mean it looks big when we're looking at these pictures. Yeah. You're like, how big is this thing?
She said slightly larger than an almond.
Larger than an almond is what we got. Yeah. Okay. Now, we're kind of stepping back in a little. So we're talking about implantation, human chorionic gonadotropin. So we need to talk about the uterine tubes first. Okay, because this is where fertilization takes place. This is going to connect, and you can see this opening here. Here's our ovary. We've got an ovulation, and it may not eject it right into the opening, the Oz of the fallopian tube. I mean, that's great if it does. But this is like a funnel that's surrounded by these finger-like processes called fimbria. And then that goes in, and this part is called the ampulla. This is the infundibulum, kind of a larger open part at the very beginning. The rest is the ampulla. As we get closer to the uterus, it's going to be the what is it called? Isthmus, the isthmus of the fallopian tube. But it's the ampulla and out here where fertilization needs to take place. See how they say day 0 fertilization? Because you've got to do a number of divisions and be at a certain stage of development. They've got you day 5, 6, and implantation down here at day 8 or 9.
So sperm has to go all the way like this? Nah.
How many,
It happens, clearly, it happens. Hang on.
how many sperm, like there's one egg most of the time.
Yes.
How many sperm are trying to get to it?
How many sperm are trying to get to them? A lot. And we'll talk about that when we get to the male reproductive system. Only one fertilizes the egg. There's going to be a number that are penetrating through the corona radiata and the zona pellucida with those enzymes that they secrete.
If there's twins, it's the same sperm.
Depends. Depends on twins, and we'll talk about
Oh,
that.
that has to do with like
Yeah.
the.
So we're all
Yeah.
winners because we all made it to
You are all winners, that's right. Okay, so the fallopian tubes, we got the mucosa muscularis, the serosa. We're familiar with the serosa on the outside. The muscularis, 2 layers, that's going to give those peristaltic contractions. Again, pulsing so that we can move material toward the uterus. Okay, come on, let's go this way. So, not only the sperm have to swim all that way, they have to swim upstream against the flow. And also, your mucosa has cilia that's beating, creating its own current toward the uterus. Yeah.
Like,
Yeah.
they're going like this in the sperm.
Sperm, you'll go away, go away, go away, go away. Yeah,
Okay, this is a weird question. But
weird
do,
question.
Like, do women like feel the muscularis pulsating during ovulation?
I don't think, if you don't feel the ovulation, you're not going to, no, you're not going to feel that at all. I mean, these are really, really small little fans kind of beating.
Is there a base and apex of the ovary? 'Cause, like, what if one of the follicles popped over here?
And again, depending on where the ovulation occurs, remember, you've kind of got a current bringing material in. So even if it's out here on the side, it's going to be going in this direction. But again, not all make it to the tube, and we'll kind of talk about that as well.
So the fertilization happens like pretty close right here. So is that what happens if like it stops like moving like this way and it starts to like day 8, you know,
Yeah.
it starts to implant? Is that whenever you have like a fallopian like birth, or not birth, but like
A tubal pregnancy? Yeah, and we're going to get to the tubals. They're ectopic.
Yeah.
They can be on the tubes. They can be anywhere in your abdomen, actually.
there was
Yeah.
one outside of the like.
Now, I didn't include this in the lecture, but there are some, okay, listen up. This is probably the weirdest thing you've ever heard in your life. And you can Google it and see it. If you have a fertilized egg that is fertilized and it does not go in the fallopian tube, and it doesn't necessarily attach but settles in your lower peritoneal area, maybe in a peritoneal pocket, it can begin to develop and go so far, but I want you to look up lithopedian pregnancy. Lithopedian pregnancy. Because what you will see are basically calcified looking fetuses. They look like the statues from Vesuvius.
My God.
Yeah, this is an ectopic outside the uterus, just one of the crazy things that can potentially happen.
Yeah, see, it's not in the thing. It's like
Is that like
Yeah.
a stillbirth thing, or do you?
Oh
like,
no, you have to have it
C-section?
surgically. Yeah, you have to have it surgically done.
Yeah, there was one that had like it in there like forever, like years.
A lithopedian like this?
I don't know.
Yeah.
Exactly.